It's great to be here, and I choose a title which hopefully spices up a little my talk, The Billion-Dollar Molecule. Before I start, I would really like to thank the organizers for inviting me and giving me the opportunity, especially you having to be here. So, we talked about chemistry already today, and today we see some very special chemistry on molecules which are extremely valuable and extremely difficult to make. So I hope I will be able, in my talk, to give you some idea of how this works, and I will divide my talk essentially into two parts. The first part gives you a general overview on drug discovery in more general terms, and later on I will show you some of my research in the area of natural product screening, which of course is something which definitely is of high interest to Indonesia, both as an industry but also as a scientific endeavor. So, the billion-dollar molecules goes two ways. First of all, it takes nowadays about $1 billion to make a drug molecule. This is the average price tag which normally you have to pay if you want to bring a chemical molecule, a normal chemical substance, from discovery through the clinics to essentially to the marketplace, to the pharmacies where you can buy it and you can use it for treating diseases. Not everybody has $1 billion, so it's a frightening number. And of course, many people are working very hard to get this number down by inventing novel scientific concepts, and that's in fact most of our work which we are doing is directed towards that particular part. The upside obviously is if you are able to make the race, to get this compound through all the barriers and bring it to the clinic and to the pharmacy, you can earn quite a lot of money. And so the average drug brings in an annual turnover of about $1 to $5 billion, depending on the disease you're working on. So it's still, if pharmaceutical companies are kind of sometimes complaining about this first price tag, you should always tell them the other one, because you can make tons of money if you're successful in that way. As a scientist, we are a very important part of it, and you will see today that it's not a single science which is doing the job, but we need sciences from material science, what we heard before, but especially also biological and chemical sciences. Computer science becomes nowadays extremely important in doing design of molecules and all kinds of studies. So I would say pharmaceutical science is a sort of integration of pretty much every science you can find in your faculty. And of course, at Leiden we have our academic center, which is trying to do that in our area. So this is in fact the benchmark. And if you look to what's really happening is that in the beginning of this drug discovery project, we have tens of thousands of compounds. That's what the typical starting point is. So we have chemists or biologists who make huge chemical libraries. And in fact, developing drugs essentially is a race to get rid of most of these compounds. Because you can never ever bring that many compounds even closely to the clinic and test it on animals or on humans. So many scientists are developing concepts to kind of filter this enormous chemical space and pick out those compounds which are really becoming, hopefully, candidate drugs. You can see a typical phase how drug discovery works. In the early stage, it's a lot of chemistry and biology. Afterwards, we go more into toxicology, so we test the safety of compounds and so on. And finally, after quite a number of years, 6-7 years, it's really a long race, we hopefully are able to bring a few compounds into clinical trials, which means we take these compounds and we go to patients and to persons to see whether these compounds are doing the job properly. And eventually, after almost 12 years, hopefully bring this one single compound to the market. So scientifically, this is really a crazy race because there is really so much money spent on compounds which will never make it. So that's not something which hopefully happens in any other kind of industrial science. 12 years, by the way, is also a very long time. If you look to turn around of other products, if you take your iPhone, for example, every year you get a new one. In 2 years you get a new design. You wouldn't want to wait 12 years before somebody invents a new iPhone. Drug discovery is a discipline which is really in for the long haul. So here's how things work. We start, of course, with humans. Eventually, it is, of course, a simplification. We bring it back to organs, so the area where diseases are really occurring. And then comes the critical part where we have to find the tissue, the cell tissue, where the proteins are involved, and I think these are, the point isn't working here. These are the molecules essentially which we want to identify. Proteins are essentially the attacking points for most of the compounds you can buy at the pharmacy. So we will try to bring back diseases to a molecular level, because the molecular level is in fact our way to approach this drug discovery process. So here we see very simplified the entire arena. So we have the protein finally identified, and then comes the chemical compound around. Most of the drugs nowadays are still small organic molecules, although we see quite a lot of developments in going into large protein drugs, but I will not talk about those today. And essentially what you then have is really that you bring this compound into contact, and I try to simplify it here. You have the protein, you have the compound, and essentially what happens is that the compound has to bind to the protein to have an action. This is a key principle which has been discovered many, many years ago, that essentially still generates a drug action in most of the diseases which we are attacking. So, the first question is, where do these drugs come from? And of course, traditionally, nature has been an incredibly important source of chemical components for drugs. The early drugs all came from nature. I would say until the 60s or 70s, probably 90% or even more of those compounds were derived from nature. And only later on, chemists in fact starting to build compounds in the chemical lab without using any kind of natural footprint. And later on, with the event of high-performance computers, we are able nowadays to design drugs behind computer screens. So I will start and go through that very briefly. So if you go to nature, if I give a course, or if I talk to students or any kind of audience, and I ask you which kind of compounds would you like, which would you prefer, a compound from nature or a compound from a chemical lab? 90%, maybe even more, of all people say, I want to have a compound from nature. And of course, this slide should show you that nature can make very nasty stuff as well. So the fact that anything is natural doesn't mean at all that these compounds cannot be dangerous. So here you see a chemical structure of a compound you can find in a pufferfish. In fact, it's produced by bacteria, and the lethal dose is about 25 milligrams. So if you go to Japan and you want to eat this fish, you take care that you go to a good restaurant. Otherwise, you have a serious risk by having a tiny spot of that compound and die from it. It's a very nasty compound, which is very difficult to detect. Yeah? What I want to show you here is that it's really a very active compound. It's a compound which has an immense strong action in our body, and of course those compounds never ever will become drug. Nobody will be able to administer such a compound to any kind of patient and hope that he or she will survive. Of course, as I said, historically we have found many useful compounds in nature, and I think the nice part is that you can find compounds in all kinds of environments. So typically in Indonesia I would say you would start with plants as a source, but nowadays, and I have an example later on, also animals can be a highly valuable source for starting point of these chemical compounds. And I have another example of a microorganism, like bacteria or fungi, can play a highly important role in drug discovery. So the reason why nature is such an attractive source of compounds is that nature makes compounds which you very difficult to make in the lab just by designing them. Nature is very intuitive. These compounds have undergone evolution for many millions of years, so you will find structure which you will never find easily in a chemical laboratory. But that's, and here's a nice example for an anti-cancer drug, vincristine, which is nowadays on the market, a very complex chemical molecule if you look to it. Not something which you would easily make, even if you would have very sophisticated chemistry. It was found in nature in certain trees, and it became a drug against cancer. But it has to be said, nature is not a very easy source of compounds. Because imagine you take a plant and you extract all the chemical compounds from that plant. So you get thousands and thousands of compounds in that extract. And then of course the big question is, which of those compounds is the active one? So you start a cycle of preparing the extract, isolate compounds, elucidate the structure, and finally go on to screening. So in other words, measure the biochemical activity. But typically this cycle has to be done many times before finally you end up with the real answer, which of those 10,000 of compounds in a plant extract is truly the active one. Because remember, if you want to take a compound into clinics, you have to describe, you have to explain which compounds it is. You can't take an extract containing 10,000 of compounds. No agency will ever allow you to do that in an easy way. So nature has also its drawbacks, like many other things as well. It's time-consuming and very complex to extract those active compounds. Not unimportantly, it's very difficult sometimes to get your hands on the sources. We don't want to harvest entire forests talking about biodiversity just to get half a gram of these compounds. So getting reliable sources is very important. Maybe one of the most important reasons why natural compounds don't make it effect nowadays is that for many companies, the time it takes to synthesize these compounds in the lab is too long. And in fact, chemists are way faster than nature in that particular aspect. That's probably the main reason why natural products in the last 20 years perhaps have gone down as a major source of new drugs. So when one thing goes down, the other thing goes up, and what went upward was chemistry. So about in the 90s, chemistry went into a phase where we were really able, by doing parallel synthesis, to really build 10,000s of compounds in a very short span of time. And that was a starting point where many companies essentially abandoned their natural product departments. I think I saw the last one at Bayer in Germany, which was maybe even nowadays. I think they are still in place, but many compounds stopped and essentially fired the entire natural product department, which is really a shame because you had extremely good chemists and biologists in there. And they did it because the synthetic chemists were able to provide them with a much more reliable and faster way to get these compounds. So then you can really get, nowadays, if you would go to a major pharma company, they would have warehouses essentially, so huge storage spaces where they store 1 million compounds or more as a source for developing novel drugs. And that's something which is very hard to compete with, unless, of course, if you look at the drawbacks. Because to screen 1 million compounds, to really take every one of those compounds and determine whether they have a certain binding affinity for that protein, which is responsible for the disease, it's a hell of a job, and it's very expensive. So I would say lately, the big companies at least, they start to shy away from these extremes in compound warehouses, and they really want to go much more into dedicated libraries which contain highly diverse compounds. And that's where nature, in my opinion at least, starts to play a very big role again. Another part, of course, is if chemists make compounds, they tend to use chemistries which produce very similar compounds as well. So the big danger is that all these companies build their storage with compound structures which are all alike. And if they are all alike, it's very hard to get them to the market, because essentially it might be even happening that a competitor already found that compound before and is patenting it, and then you are gone. So it's really a very high risk of having me-too compounds, structures which are very predictable, not diverse at all. So about 10 years ago, with the event of high-performance computers, people start really to think about calculating chemical structures. And not synthesizing these compounds anymore, but essentially doing what Katja described to you before. Take big computers, put in protein models, and then try to find, by simulations, compound structures which have a good fit, essentially in the same way as she described it for optimizing enzymes. So the idea is that you use a computer, you need the protein structure, and you go through billions of compounds, even if you like. It still takes a lot of time, and then isolate a few of those which you can use in the chemical lab to build them. What you need are, of course, strong computers. Without that, you shouldn't start. And what you also need, and that is immediately the biggest drawback, you need protein structures. And nowadays we found those structures for quite a number of drug targets, but not for all of them, and certainly not for the most important one. I'm going to show you one later on. So here, a big drawback for doing only computer-based drug design. But once you have all these compounds in place, then of course the question gets, what is ideal drug? And here I gave you a big overview on what has to be in place to get these compounds into the next phases of drug discovery. a lot of different aspects. I don't want to go into them. I will later on come back to innovativity and to natural compounds. So the ideal drug, of course, is very effective and it's very selective. It only treats the disease. It has a desired response. So if you administer it, it's curing the disease, and of course it should be safe, even at very high concentrations. So if you take it, you don't die from side effects, the last thing you want to have. And of course you don't want to have any secondary effects like habit forming. So there are a huge number of demands on these compounds nowadays. And if you see what is really happening for the ideal compound, ideally you would like to swallow it. It should not be administered intravenously, but you want to take it as a tablet. It should essentially reach the site of action very easily, which is very hard in the brain. As I showed you here, this is a very big job to get compounds selectively into the brain area. It should reach the site of action. It should bind to the protein, and essentially it should also be excreted again. So if you take a drug, it should never stay in the body indefinitely. It should get out again. So as you can see already, we have many demands on compounds which might become drugs in the long run. And of course, that should be said, many compounds which chemists in nature are designing are failing. And they are failing after a lot of research has been done by chemists and biologists as yourself. So as I said, many of these trials of these tests only take place after 6 or 7 years. And then it can happen that a compound is not effective. It can happen that it's toxic, it has poor properties. A competitor has found it before you, which is really a bad thing to happen, or you can't patent it because somebody else has done it. So a lot of things can go wrong and do go wrong. And I always have that example here of a compound which was developed by Merck about 10 years ago. It was an anti-pain compound called Vioxx. You might have heard about it. It was deemed to be a very successful compound. For Merck, it was really the compound which was their biggest hope in really staying in the big race of staying in the big pharma compound. It went through the entire 12-year cycle. So it went through into clinics, into every clinical trial. It passed the clinic. It went to the market. And only when this compound was in fact available in hospitals and in pharmacies, then suddenly a bad thing happened. People died from a heart attack. And then of course, people first of all thought it was a side effect, but essentially, in the end, Merck had to take this compound from the market again. This is the major accident which ever can happen to you. After you spend $1 billion to develop that compound, you don't want to take it from the market. And the reason is it has a biological source. This is the enzyme which was the drug target, called COX-2. So the compound is made, the Vioxx was a COX-2 inhibitor, so a compound which bound to this protein very tightly. It had very good pharmacological properties. People liked this compound. It had, it was a great painkiller. It had a very low risk of ulcers, which was a problem of other painkillers. So when this compound came to the market, many patients had very high hopes that this is the painkiller of the next generation. But unfortunately, it turned out that this compound also led to clot formation in blood, to high blood pressure, and essentially to heart attacks. So the good side and the bad side was in fact cancelling each other. The compound was taken from the market, and after it was taken from the market after a few years. So this is, you can read it here. Many patients died already due to heart attacks. So very hard lesson to be learned. So this brings me back to my second part of my talk, coming back to nature and how can we find drugs in nature much more quickly. That was my field of research, and I want to show you two examples in the last part of my talk from microorganisms and animals. And of course, as I said, if you want to have compounds from nature, innovativity is the biggest selling point. You will find structure which you will never ever find easily in a chemical lab. So, and I'm glad Katja already introduced the enzymes, because my first example also starts with enzymes, with very specific enzymes, in fact the ones which we have in our liver. So we had the idea that if you take liver enzymes and we use them to modify compounds, essentially it's metabolism, that the new compounds which we are forming in this way are much more beneficial and much more better drugs than the parent compounds. And we went into this area, and of course we collaborated with many colleagues from the biological field. They built, they found bacteria which were expressing these enzymes. They could make a lot of mutants, which essentially helped us to build all kinds of chemical structures from certain starting points. So it's really a combination of lead discovery and biosynthesis using enzymes to increase the diversity of chemical compounds. So we use natural compounds, natural enzymes to increase the diversity of chemical components. A very powerful field, in my opinion. But, and this is my field of research, it's very, very tough if you start here to identify all those compounds you are making. So what you do is you take a starting compound, let's say the compound here in the middle, L1. You take these enzymes and you let them move in that jar. So what they're doing is they build new compounds. And they make derivatives of this parent compound. But you have no idea whether the new compounds are active or not. You're ending with a mixture of active and non-active compounds. And then of course we wanted to build technologies which can do essentially the entire job in one go. Which means separate all these compounds in first place, screen them and determine their chemical structure. This was the field I was working on. So this is again a very tough job. Hopefully you have seen these kind of technologies before. You will find them in many, many labs nowadays. It's called liquid chromatography, mass spectrometry. This one here is a separation science here. So we use separation science to separate out all the tenth of compounds in our sample. We use mass spectrometry to identify those compounds. And what we brought to the table is in fact essentially the biological dimension. So we took the drug target itself, we built a screening assay, and we ran the screening assay in parallel to the chemical analysis. And this helped us, in fact, to do the entire analysis of this complex mixture in one go. And I just show you how this works. So we have to build chemical biological assays, and here you see a very easy one based on fluorescence. So essentially we take the protein target. This is our compound which we want to test. We add a fluorescent compound to it. Now, if there is no active compound around, you get this nicely fluorescent complex here, which we can measure by fluorescence spectroscopy. But if we add anything which is active, anything which binds to the protein, which is non-fluorescent, it is blocking essentially the binding sites, as you can see over here. So you get a decrease in fluorescence, which we can easily measure using a chemical detector. So here's a system which we ended up with. Looks a bit more complex than it really is. So you see here the chemical dimension, separation science, where we separate all these compounds. We have a splitter over here. So it's a flow system where one part goes to the mass spectrometer. Here down we get the chemical information of this natural extract or the extract of bio-derived compounds. And in parallel and at the same time, we do the biochemical assay, which we need to tell us where the active compounds are. And then in the end, you only have to correlate these signals over here and derive the chemical and the biological information in one go. We did it with this system. So here you see our starting compound. It's a steroid which binds to the steroid receptor. We take this lead compound. We use the P450 enzymes to make a lot of metabolites, which essentially can be active or not. And we use our system to do the analysis. And here you can see what comes out. This is the chemical dimension. So this is what you will see if you just use LC mass spec in your lab. And by adding this biochemical assay, we do an overlay of the biological dimension. And then just by naked eye, you can already see the parent compound here. You can see one active metabolite because it gives a signal both in the chemical and in the biological trace. And over here you can see that in this process we made another compound which we easily can identify, but it doesn't have any activity because it doesn't decrease the fluorescence over there. And that system we use for doing combinatorial biochemistry. So we took many compounds, incubated it with all these mutant enzymes, and then you get these kind of pictures. Here you see a native enzyme from a rat, and if you go through it, you see the parent compound, which is active, of course. You see many compounds which are metabolized by the native system, so the system which mammals have in their body. And of course, the most exciting part is over here. Here we find novel metabolites. These ones are made by rats and by the mutants. And last but not least, you see compounds which are made only by the bacteria. And of course, those compounds are the ones which were our test compounds to go into further stages of drug discovery. Now, as I mentioned, this is very combinatorial biochemistry, and I wouldn't want to leave you without having to show you a real example from nature. And this is work which a colleague of mine has done in Amsterdam. He was looking into venoms from snakes. Now, I wouldn't normally consider snakes as a real source of compounds which you want to put into a human body. Ideally, that is not the case. But snakes have venoms which have highly, highly active compounds. So the idea was we take those compounds, and if we find that we modify them later on, make them less toxic, and use then those compounds as drugs. And there are quite a number of examples already on the market. So my colleague, he was interested, Jeroen Kool from the University of Amsterdam, to start with snake venoms, identify the active compounds, and then essentially take them further in the stage of drug discovery. Now, here we were looking to brain diseases. We looked to ion channel proteins. That is the nicotine receptor, which was our drug target, a highly complex protein which is in the membranes of various tissues. And essentially, we were not able to isolate this protein easily to use it as a drug target in our assay systems. So we tried to mimic it, and here again, nature came around. Scientists from other groups, they found the same protein, the same binding site, not in a place where you would typically not find it in a snail. And this so-called acetylcholine binding protein had identical properties to the receptor in our body. So rather than using this very complex membrane structure, which is very difficult to isolate, where we don't have any kind of structural information, they use this protein down here to build the assay exactly in the same way as I showed you before, and run snake venoms through this assay. And this is just using exactly the same system as I mentioned before. So we separate every compound from the snake venom extract by using liquid chromatography. We split the flow behind the separation column, and one part of the flow goes to a chemical detector, in this case again a mass spectrometer, which gives us the chemical information on all these different types of compounds, and the other part goes to the biochemical assay which we run in parallel on the basis of this acetylcholine binding protein. So rather than using very complex membrane proteins, we took the easier one. And here you see a nice example from an extract from an actual snake. I don't even dare to pronounce the name. I'm not a biologist, so hopefully some of you will be able to do it. I will test with Fiat later on whether he knows that snake. I didn't do it. But the chromatograms are very easy to interpret in the end. Up here you see again the chemical information. So you see many compounds which are present in this extract of the snake venom. And it would be very hard if you would like to extract them and to fractionate them and then do the test in any other way. By doing it online, by doing a biochemical assay in parallel to the chemical assay, we were able essentially to explain the entire story here, because you can see that here we have some peaks of totally new compounds. Nobody ever has reported on those compounds before, so they are investigated. We saw other ligands which were reported before, and we saw other toxins which were reported before. So with one single chromatogram, if you look to the time axis here, it would be about half an hour. We could essentially solve the entire puzzle of a highly complex biological problem, namely identifying bioactive compound in extracts of snakes in this particular case. And I truly believe that by combining technology together with chemistry and biology, we will be able to again use nature as a very valuable source of novel drug compounds in the future. And that's of course something which is extremely important also for a country like Indonesia. So in conclusion, I was able hopefully to show you a bit about the complexity of doing drug discovery, of finding novel compounds which can be put into the clinic. I think the major take-home message here is, and it's really a hopeful message, that it requires scientists from various different disciplines. You cannot do drug discovery development by just one single discipline. It requires chemists, biologists, computer scientists, mathematicians, everybody which might be here in the room will find a job in this particular area. Pharmaceutical companies and universities are hiring PhD students, postdocs, and staff members from various disciplines. If I just look to my own institute in Leiden. I can also assure you that we still have a long way to go to find drugs for the most complex diseases nowadays, because they have a very complex biomolecular background. And as I mentioned, nature, in my opinion, is a very valuable source of bioactive molecules, and hopefully countries like Indonesia will be able to also go into this endeavor. It requires a quite significant infrastructure, but by collaborating, for example, with other scientific groups in other countries, you might be able to combine all the expertise required to do successful drug discovery. And with that, I would almost like to end my talk, not without showing you this particular picture here of our institute, which is located at Leiden Bioscience Park, which is the largest cluster of biotech and pharmaceutical companies in the Netherlands. Our institute, the LRCDR, is right in the middle. Our students, we have plenty of those, they find jobs in all kinds of companies, both pharma companies but also biomedical companies. And it's a unique environment for collaboration in the future. If you have any further questions, here you see my email address, and of course, I thank you very much for your attention.
Thank you. Thank you, Professor Hubertus. I think this is really mentioning about this, the need for collaboration within multidiscipline, because one discipline cannot be resolved for the drugs discoveries. Also, it's very interesting between the synthesis of natures and computer-aided drug design. It's really very new things. And how that's really the microbes find an animal, especially in Indonesia, with a high biodiversity, can be really finding of new drug here, especially in trying to find the bioactive compounds. So I think this is really a very amazing lecture. And anybody want to give any question or yes, I can see the guy there.
Okay, thank you for the opportunities. My name is Esti. I'm from Biological Department from University of Indonesia. I'm really interested with your presentations. I have two questions basically. My first question, I do agree with your presentations that Indonesia have a lot of biodiversity, that is still high possibility to find the drug discoveries in the next futures. My first question is, sometimes the challenge in drug discoveries is from the nature's plants, animals, microbes or something, is beside how to take it, the place, how it's very hard to take it. Sometimes the amount is also limited. How you enrich the bioactive if already found, to enrich the amount from the bioactive that already found? As I know, mostly we just take the gene and transfer to the bacteria, and then we harvest from the bacteria. Is there any ways to enrich the bioactive compound? That's my first question. The second question, I'm just curious, some of the disease have a protein target on the membranes so fast to mutate, so we cannot predict if we find the drug. Sometimes the disease already mutate and then it's already different again. Through the computer designing, is it possible to predict the mutations of the protein target on the membrane, cell membrane, so that we can make the drug before it's already shown? For example, like the swine flu, the avian flu or something. Because mostly when the drugs is already happened, we try to find the drugs, but when we already find the drugs, the new disease from the another mutation from the disease already shown. Is there any possibilities to predict from the computer already? Thank you.
Okay, any other question? Yes, Prof.
Thank you very much. My name is Tony Sumariada. I'm from Bogor Agricultural University. Actually, I'm a physicist, but I have my colleague in Bogor Agricultural University to run the program for the design, like running the autotropina and then checking the toxicity. Actually, I use a free software like Prof. But can you tell me the state of the art of the program to predicting the toxicity? Because this is, I think, the main problem in when you do the drug design. There's a lot of program that you can predict how such a compound inhibit the target based on physics equation, the Gibbs-2 energy and so on. But predicting the toxicity, I think it's still a problem. So can you tell me about the state of the art right now? Thank you very much.
Well, I think I start with the first question because it's a very important one. Essentially, once you find an extract which contains active compound, the question was how can we essentially enlarge that compound without cutting down a huge number of plants or trees or whatever, or catch a lot of animals. I think about 10 years ago I would have said this is probably one of the main reasons why it's so hard to do that. What has changed in the last 10 years is that the instruments which we have at our disposition for structured elucidation have become much more sensitive. So if you look to NMR, which you have seen before today, you look to mass spectrometry, I think nowadays we are able to elucidate structures on using much lower amounts of compounds than 10 or 15 years ago. And that what then afterwards will happen that you don't probably would go back into the biodiversity environment and collect more of it, but really try to build it in the lab. Try to find synthetic ways to reconstruct the same compound using traditional organic chemistry means. I would say this is probably the easiest way to go forward. You might need a bit more compounds if you have very complex molecules which are chiral and so on. But ideally, let's say if you want to stay in this time race, you should be able to synthesize the compounds afterwards in the lab. I think nobody would really want to extract kilograms of compounds from the forest. That would not be a way to go. So really the hope and the benefit is that the instruments nowadays are so sensitive that you can do that job, hopefully with much smaller amounts. Regarding your second question, I must admit that I'm not an expert in the area, but of course the question is very important. If you have diseases which are based on either protein or viruses which are mutating very quickly. In this particular case, I would say you would probably not want to go into small molecule discovery, but you would go into immunotherapies or other kind of biopharmaceuticals which might attack the problem at the source and not at a particular protein level. But again, I would not say that I'm an expert. I can easily bring you into contact with these experts, but I'm not one myself. And the same is true, unfortunately, for the last questions. I'm not a toxicologist, but we have very strong toxicology groups, both in Leiden, also in my former institute in Amsterdam, which might be able to answer the question about the latest software which is predicting. And we have both groups are working on predictive toxicology, because exactly the same is true then for other areas of drug design. You want ideally a computer program which helps you, if you have a structure, to predict things rather than going to animal research, which is also something which is really declining, at least in the Western world. We are not allowed anymore to do extensive animal studies. So predictive toxicology using computer programs is a key aspect to have in place. But the exact software, which is the best one nowadays, I would rather not answer that question. I might give you the wrong answer, and I don't want to do that. Send me a mail, I will take care that you get the answer.
Yes. we have Professor Usman Sumo, really doing research in this poison and snakes and drug discovery. If you want to have something to say.
Okay, thank you. If it is, let's say, a protein, peptide, small molecule, so to enrich this compound can be used genetic engineering tools. Okay, but if it is protein big enough, so it's difficult for us to enrich the one by using this genetic engineering. Okay, but if it is a natural product, it's not. I mean that peptide-based drug. Peptide-based drug, it can. Then the other one is the toxicity. About the toxicity, the same thing like the others. We can wet experiment, we use it and develop software to do that one. Then we can use it to predict toxicity. That's the point.
Thank you, Professor.
Thank you.
Any other? We have one more. If you anything that want to say? If not, I can ask you about this. Indonesia has 500 tribes, and each tribe has different kind of medicines. Which is faster with using the computer aided drug target, or using the traditional way of doing things like ethnobotany, ethnobiology, ethnomedicine? Then you can screen those instead of randomly check all these species. What do you think about that?
Well, first of all, I think it's of course scientifically it's a very complex question, because these traditional medicines are quite likely made up by many active species. Quite likely it's not a single compound which is taking care of the medicinal action of these extracts. And so to elucidate many active structures in these extracts, I would say it's a very tough job. But frankly, I would say leave them where they are and use it as a whole product, rather than trying to figure that out. I think there are people who are trying to do it, but I, quite frankly, because I think even drug agencies are not too happy about combination therapies, combining 2 or 3 different drugs. They are very reluctant nowadays to approve this kind of medicines. Also, I would say rather leave them where they are.
$1 billion.
Yeah.
That was really
Yeah.
the key message.
I should have put the figure a bit down, not to exaggerate your hopes.
All right.
There are very few companies who earn $1 billion.
Yes, because in biology department, Bioscience, you can mention about this, because we had a group they call the bioprospecting. So we have a group that research in the Faculty of Mathematical Science on the bioprospecting. So I think we are interested in this kind of collaboration in the future for more on biodiversity screening for
we certainly are interested in that. So again, it's from an interpretation point of view. So if you have, let's say, 10 or 15 active compounds which act on different proteins, even, that to translate this into science is I think beyond our means, as far as I can tell.
Okay. Thank you very much. I guess that let's give a applause to Professor Hubertus.
Thank you very much, Professor Dr.